I used to be known for my puns. I was the king of dad jokes. I was known for anytime someone said, "Oh my gosh," I would say, "You have a gosh?" I've always wanted to know what one of those looked like.
So dumb. I love it.
Welcome to Absolute Genius, a new podcast series from Thermo Fisher Scientific. I'm Jordan Ruggieri.
And I'm Cassie McCreary, and today we're taking a deep dive into the world of microbial ecology with two geniuses, Dr. Brandy Keel-Reese and Lydia Hayes.
Brandy is a senior marine scientist at the Dauphin Island Sea Lab and an associate professor of marine sciences at the University of South Alabama. Her work explores the microorganisms that survive and thrive in extreme environments, like the deep ocean. Lydia is a PhD student at the University of South Alabama and works closely with Brandy on several amazing projects.
So grab your headphones, a roll of duct tape, a toilet brush, and a cool refreshment as we MacGyver our way through this fascinating conversation.
The toilet brush comes in handy, we promise.
My name is Brandy Keel-Reese, and I'm a geomicrobiologist, which just means that I study microbes and microbiology in an earth setting. And in this case, things that live in the bottom of the ocean, things that live in the crust below the sediment of the ocean. And that includes microbes like bacteria, archaea, and even fungi.
Hi, I'm Lydia, and I am a geomicrobiologist in training. I'm a PhD student here at the University of South Alabama, and I am interested in microbial life in extreme environments, what they do, how they survive, and how they interact in the environment.
Amazing. I have to say, too, that I know you're looking at the Mariana Trench and some of the mud volcanoes there, and also had some expeditions to Antarctica. We'll talk about some of that as we go through as well. What questions generally are you trying to answer as you study these microbes?
We have a lot of different research questions that we're addressing, but overall, when we look at an extreme environment like the Mariana or Antarctica, we're looking at what kind of organisms live there, what they're doing, and how they're able to survive in those extreme conditions.
Just in general, we want to understand survivability on this planet, and single cells use unique genetics to survive. And so we want to look at it from a genetic standpoint, but we also don't want to leave out those that we can grow in the lab as well. So we use culture-independent and culture-dependent approaches.
With such tough conditions, there must be ways that these organisms kind of adapt and compete with other organisms that are there. Maybe low biomass, low oxygen. Is this the case? Do you see these with these organisms?
Well, exactly. Each site is unique in terms of what is considered extreme. Extreme can be, low oxygen or no oxygen, or extreme can mean, high pressures. It also can mean high or low temperatures. It can also mean nutrients, right? So the lack of carbon, the lack of nitrogen, the lack of, other, micronutrients that are needed for survival. And what we're trying to understand is, some of those responses to survivability. To anthropomorphize a microbe or give it a human characteristic, we can think about this as a game of Survivor, in which you have microbes that can adapt or evolve in response to their environment. In other words, can use the nutrients that are available or use, the environment that's available and can thrive in that situation. Other microbes can't. And so those that can evolve or that can adapt will survive and will even thrive. Another choice, if you will, or another option is that they can just go dormant. They can be inactive, or it can actually, build a spore, create a spore around that cell. So then they just shut down to just the most basic genes or most basic needs that are keeping that organism alive. These are kind of like zombie organisms just hanging on there in some sort of purgatory. And then the third, choice, if you will, it's to create secondary metabolites that can disable or even lyse, meaning kill your neighbor. So you can, at that point, use your neighbor for its necromass. It's a little bit like cannibalism, where one cell is eating another cell, but they're very, different species, if you will. Through the production of these secondary metabolites, which are antibiotics, they can consume their neighbor and, or even at least disarm their neighbor so that they are no longer a competition.
That is incredible. I mean, you did mention a little bit of, they don't know if they're alive, they don't know if they're dead. I can just imagine that can actually cause a lot of issues when studying these organisms. You don't actually, or may have trouble figuring out which ones are doing a better job at living if you have trouble identifying in the first place if they're alive or dead. Is that something that you also run into regularly?
Yeah, absolutely. So bacterial cells and archaeal cells, for that matter, really exist on a continuum. Live and dead isn't as clear cut as it is in our eukaryotic world. A cell can exist in various forms of damaged states and even in dormant states. So we use several different context clues to try to suss this out. DNA is only one of those, and DNA is like your library. We're not reading everything all at once, all at the same time. We just know that it's contained in there, that information. And if we take out a book and we open up a page, that's like our RNA that's being expressed. So we can start expressing those genes. And we know that if an organism is alive, that it's expressing genes. So it's reading the book. If it is dormant or if it's dead, the books are put away. The information is still there, but it's not being read actively. Other levels of activity include, proteins or proteomics or even metabolomics, in which we're going to go ahead and start reading the page. And so reading the page and, garnering that information, that's like proteomics. Then when we start, maybe we're writing down and we're going to copy every word of that page, and then we'll have a copy. That's our metabolomics. So we go through these different processes to try to understand not only, who is there, that's the DNA, what are they doing, and then also what actually happened, and that's our proteins and our metabolome. Putting all those pieces of information together, we can try to help us understand the microbes' survivability.
Are there complications looking at all of these different, if you're looking at DNA, you're looking at transcripts with RNA, you're looking at the proteins as well. Are there complications with going down, drilling in the soil, bringing samples back up, and then having good sample to study from?
Yeah, contamination is definitely an issue. Also, preservation of samples. We're taking them from so far below the seafloor. When they come up, you have to process them as quickly as possible to get them preserved before you can actually be done with that sample for the time being before it gets back to the lab to be processed. So we store everything for DNA and RNA extractions and analysis in a -80 freezer on board, usually on the ship. And you want to get that processed as soon as possible because RNA and DNA does degrade fairly quickly. And also, you want to know that the activity in your sample is being preserved at the state when it was collected, rather than having those organisms continue to acclimate to their new environment. If you're taking a sample from really high pressure and you're bringing it up, you want to preserve it as fast as possible before those cells could either lyse due to less pressure, or activity could continue and it might not be the same as it was when you collected it. Contamination is also a large issue when we're dealing with our samples, because you want to know that the RNA and the DNA that you're collecting from your sample is from that environment and that it's not from ambient sources around you.
Yeah, that was going to be my next question. Is especially with the Mariana samples. I mean, you're going so far down, you have to bring that sediment up through all of the different levels of the ocean there. So can imagine the difficulty of trying to eliminate some of the contamination that's coming from just bringing the sample up out of the ocean.
The nice thing about what we were doing at the Mariana, of course, using remotely operated vehicle JSON.
Oh, didn't even think about that. That's amazing.
Yeah, so they're able to, you have your eyes and hands, I don't know, like a drone, an underwater type drone. And so they filtered a lot of water on the seafloor In order to collect these samples, we use what we call a CORK, a Circulation Obviation Retrofit Kit. And this is a subsea floor observatory. It extends hundreds of meters below the seafloor into the basement, in which there are tubes, like a straw, like a sipper, that extends all the way down into the basement, comes up onto the seafloor. There's an ROV platform, and just starts, hooking up to those, to that end of that straw, if you will, and then filters on the seafloor. And then all you're bringing up is that filter itself that is then, closed off.
That's incredible. I falsely in my head, I had this vision of Lydia just standing on this, 2-mile long drill on a boat, trying to go all the way down to get that. But that's incredible. Another question: if we're looking at things that are so deep down, so far away, why should we care about these microbes?
So microbes are the most diverse entity on our planet, most diverse organism on our planet that are responsible for recycling Earth's nutrients, recycling Earth's carbon. Without microbes, we wouldn't have air to breathe. We wouldn't have food. Our food is digested by microbes. I mean, honestly, they touch every aspect of our lives. In trying to understand not only how life has evolved on our own planet, we can also begin to understand what life might look like on other planets, both past life and also life that might exist currently. Definitely is important from an esoteric standpoint, but also from a practical standpoint of, what is recycled among our nutrients, also how harmful things are mitigated through bacterial respiration, bacterial activity. That's what we call bioremediation. And also from a basic standpoint, what kind of understanding survivability of microbes in extreme environments has given us things like, the opportunity to seek out new natural products and antimicrobials or antibacterials.
Yeah, why are microbes important? They are also one of the largest biomass categories on the planet, so they're definitely significant. They're very diverse, like Brandy said, and understanding microbes really helps us understand how life is recycled, understanding how the limits of life exist when we look at these microbes in these extreme environments and how they adapt to survive, or how they are there at all, can really give us insights onto how life formed here on Earth and potentially how we might find it in other places. We also get to understand new resources, potentially new antimicrobials that we haven't gotten resistance to yet, which is definitely important for the human aspect of things.
Are there any potential climate impacts to your studies that you're conducting as well?
Yeah, definitely. Microbial communities play a large role in nutrient cycling or biogeochemical cycling, whether that's carbon, nitrogen, sulfur. All of these types of elements need to be recycled, and carbon cycling plays a huge role in what we think of as climate change. And so how CO2 is produced or sequestered through microbial respiration or biomass definitely plays a large role in climate change.
So what methods are you using to run some of the genetic analysis? Do you do sequencing? Are you using qPCR, dPCR? What kind of methods are you employing?
We do a combination of all of those, from whole sequencing approaches, shotgun sequencing, if you will, next generation sequencing. From there, if we want to look at specific genes or specific transcripts of interest that might be associated with a particular metabolism or a secondary metabolite, something like that, then we'll go into it further with quantitative PCR or digital PCR. And then also we take, some of those and we try to grow them up in the lab. But of course, we know that you can't grow everything in the lab. We can't replicate the environment exactly. And, you do the best you can, but you're lucky if you get just a few percentage of what's in the environment, growing within your lab. So yes, we take sequencing, both the DNA through metagenomics, the RNA through metatranscriptomics. The quantitative PCR allows us to quantify, how active a gene might be, maybe through looking at its gene expression, doing qPCR on the transcripts themselves. And we often run into the problem of that we have such low biomass environment that we have to look at it a little bit more specifically. The qPCR is a great tool. If you have 1,000 to 1,000,000 copies already, then it's great. It's in its sweet spot. But if you're starting out with only 1,000 cells per gram of sediment, then you're only going to have maybe tens of copies or just single copies of that particular gene or transcript. So that's where digital PCR is much more useful, and we're able to use that to get very low detection, and that technology has enabled us to go further within the analyses.
That's excellent. We love digital PCR here at Absolute Genius, so it's great to hear that you're able to employ it and looking at some of those really rare targets. Do you use digital PCR in particular do you look at particular genes? Are you trying to quantify genes of interest, or exactly how do you kind of employ that in terms of the primers and probes that you're developing and things of that nature?
From project to project kind of varies how we use digital PCR. But from my perspective as a researcher, I'm really interested in specific nutrient cycling pathways. In the low biomass environment, you won't have very many transcripts of particular genes if you only have one cell. We would use digital PCR to follow a particular gene transcript pathway for nitrogen cycling or sulfur cycling and look at those specific genes.
Being a scientist has its perks. The Thermo Fisher Scientific Aspire Member Program is our way of supporting your commitment to science. Our award-winning Aspire Member Program is for customers at all stages of their careers.
You help advance scientific research with Thermo Fisher Scientific products, so we designed a rewards program to show our appreciation of you. Get rewarded for your contributions, because being a scientist is not just a job, it's a lifestyle. Getting started is as easy as creating an account, earning points on eligible products, participating in activities for bonus points, and receiving rewards.
That's right. Redeem points for really cool, science-inspired rewards, accelerate your career with virtual trainings, and try free products for experimentation. Visit thermofisher.com/aspire to learn more today.
Let's get back to our conversation.
Welcome to Cassie's Career Corner. Laser beam noises, confetti cannons, party horns. Anyway, I digress. So we've talked a little bit about these expeditions and everything. And what is the day-to-day like when you're on these trips?
The neat thing is that, we can go out for, 2 months at a time, or, give or take, a couple of weeks here and there. And you can collect all your samples at once and then get back in the lab and, work on them for the rest of the year. So that's the benefit, but that's also the drawback too, is that you only have 2 months to collect all your samples at once, and then you've got the rest of the year to work on them back in the lab. And that if anything goes wrong, while you're out at sea, weather doesn't cooperate, mechanical failures, that's your one chance. So you have to be a little bit of MacGyver. And then, at the same time, be very flexible, flexible with your environment, flexible with your colleagues. It's a fun experience. I will admit that it's a lot of hard work. You don't have breaks. You're essentially on mentally 24/7 for that period that you're out there. But it's fun. It is just so much fun, and it's so peaceful just to see the world from a whole different perspective. And you just feel very, very proud, but insignificant at the same time, because the ocean is a big place with a lot of power, and you are a small player, in this big, wide world.
My trip to the Mariana, we were out there for 27 days on the boat. It was the most exciting and also the longest 27 days of my life, probably. Our sampling was round the clock, 24 hours. You just have so much adrenaline because you're in a cool place. And like Brandy said, you really have to learn how to go with the flow. Sometimes you think you have everything planned out and something doesn't work for you, and you have to figure out either how to move on or how to fix it. And you only have what you have on the ship with you. You can't magic a piece of equipment to you to make sure something works. And then also sometimes you find something that you hadn't intended you wanted to sample, and then you have to find a way to sample it. Like, for instance, we had some really cool biofilm growing on one of our corks, which was the straw-like structure that Brandy talked about earlier. And we wanted to sample it because it was really cool, but it was on kind of a PVC pipe kind of thing, and we had to be careful about our method of sampling affecting the structure. So we sent a toilet brush down, and the ROV collected it in its claws, and it just scrubbed the side of this pole with the toilet brush and sent it back up. And like, you have to be able to think of innovative things like that.
I always prepare my students for the what ifs, the incidentals. So we have, I tell everyone, bring duct tape, bring electrical tape, bring zip ties. When I hire students, I want to hire them for, their creativity and for their adaptability as much as I hire for book knowledge. The book knowledge can come. Common sense and creativity and flexibility, those make a scientist, I think, much more than, even what is, written in a textbook.
I kind of can't help but wonder, how did we get here? For both of you, this is a pretty, I would say, niche area, right? So how did we get here?
That meme that you sometimes see, with the way that we think science goes from point A to point B, and the reality of it is that it's a circuitous path that goes through loops and turns, and and then you get there. Well, I would venture to say that career trajectories are exactly the same way. For me, I started out as pre-med, and I decided that I didn't want to work on humans. So I took a geology class, and I took the geology class because it offered a field trip to the Grand Canyon, and I thought that would be great. And I just fell in love with geology, with the earth. I've always been a bit of an environmentalist, even back into my elementary school days, save the planet, save the earth. It wasn't until after college when I first became a consultant, before I even, went back to graduate school. I became an environmental consultant, and I was introduced to the world of bioremediation. In other words, using microbes to clean up our planet, to clean up our spills, our messes, our accidents. And it combined the interesting parts of, microbiology from that I enjoyed in the pre-med days to the soil, the environment that I came to love in my geology undergraduate degree. Bioremediation was just my first introduction to microbes, not only existing, but also manipulating and changing their environment. And from there, from my consulting days, I met some PhD researchers that were on various projects that I was working on as a staff scientist and a staff geologist. And they actually told me how to go to graduate school, which is not something that I learned or came by naturally. In other words, I applied for graduate school once and I got rejected. And I got rejected because I just applied just the same way you would as an undergraduate. But I never reached out to an individual researcher and said, hey, I like what you do. I want to be in your lab. Do you have room for me? I reapplied for graduate school, and I reached out to those that had, interests in both microbes and biogeochemistry, and then the fit was natural from there. It didn't have a rejection anymore. I had, resounding acceptances. And so that's where I started to, really explore microbes in the environment. And from there, I just kept moving further and further offshore. inland lakes to rivers to offshore, to onshore to offshore, and then just going deeper and deeper until I found myself in extreme environments.
I had a very different experience getting to academia than Brandy did. I definitely thought I was going to be a professional musician when I grew up.
Oh, okay.
I played piano since I was 8. Grew up in a very musical family. My dad played the cello. My mom plays the guitar, and she's a vocal teacher. I took every music class I possibly could in high school, and then went to college and pursued my future in music, and then realized that maybe I wanted piano to be a hobby and not a career. So then I kind of thought to myself, let's regroup. From there, I went back to community college and I decided maybe I was interested in human health. So I took some pre-med prerequisites, and the very first one that I took was a introductory to microbiology class. I just loved it. I realized that microbes were just so intriguing and all of them are so different and you can learn so much from them. So I went on to my 4-year institution and as a microbiology major. And in order to graduate, you have to do research experience for that program. And so I reached out to some researchers at the university and found a lab that was doing geomicrobiology and did my first semester there. And I stayed there for the last 2 years of my undergrad and just loved it. And from there, I was like, well, maybe I am interested in grad school for environmental science, and kind of stepped away during my master's from microbial research into chemical nutrient cycling in coastal wetlands. From there, I graduated and I took a break from academia and went to work in industry for a little while. And then I get a phone call out of the blue from Brandy, and she's like, I have this project that I think you'd be really interested in, which happened to be this Mariana project. And that was kind of my squiggly route to getting here.
It's a small world.
Yeah.
It really is. So I know her former, undergraduate advisor. Geomicrobiologists in extreme environments is a very small world. And also when you see talent like we saw in Lydia, you don't let that go. You don't let that pass you by, at least without making sure that their goals and they're where they need to be, and so hence the phone call to Lydia. And I said, are you still interested in pursuing, a PhD? Because we've got this grant that's funded, and I think you'd be perfect for it. Yeah, you don't let that slip through your fingers.
That gave me the warm fuzzies. I now want to bring us to our first ever Cassie's Career Corner Challenge. So congratulations, you guys are the first participants.
I'm a little nervous.
Isn't that great? I know. Sum up for us and our listeners in 5 words or less your field.
You're asking a scientist to be
I
short-winded.
I have to think about this. I'm over here counting words.
Once we think of our words, we can say them all together as if this was recorded right for the first time. Microbial survivability,
adaptability,
microbial survivability in extreme environments.
There you see? Look at that. Teamwork makes the dream work. Well done, you. I have no prizes for you, but consider yourselves blessed. Okay. I like to do this every episode because we love consistency. I want to hear from both of you, please, your best or proudest lab moment or research moment, what have you, and also your biggest oops or oh no.
There were a few very proud moments.
Okay.
The toilet brush was definitely a proud moment.
Mm-hmm.
Another proud moment would be we were having some problems with our sampling method for fluids, and I came up with an idea to send a really long tube down into that straw, and we got very pristine samples from that, and I was very proud of that idea because we kind of thought at one point that some of our samples were awash. Sometimes the simplest solutions are the best solutions, so that one was a really proud moment as well. My biggest oops. I haven't had any that were super detrimental yet.
Okay.
Knock on wood.
Well, that's good.
But,
yeah.
I think probably my biggest oops was one time I was mixing some really high concentration sodium hydroxide and didn't know that it created a very exothermic reaction when you mix it all together. And luckily I was doing this under a hood and I was wearing all my proper PPE, but I mixed it all in and the exothermic reaction bubbled and it exploded in the hood and just shot high concentration sodium hydroxide everywhere. And now I know that you add it in a little bit at a time and you mix it very slowly. Otherwise, it is bad news bears.
The funny thing is that we have that exact same story, that we both did that exact same thing, during our, early career stages. We've joked about that before. We learned the same lesson, not together, obviously, separated by, maybe 15 years probably, but that I did the same thing with the sodium hydroxide, created a pressurized, cap, of a bubble that underneath it, start to swirl it and it, explodes. My proudest moment absolutely comes when I graduate students or watch the students accomplish something big that was, very challenging, or overcoming obstacles that might be in the lab, or their challenges, out at sea, or just in general, once they publish, that in of itself can be, a challenge in overcoming, your writing fears, or your writing, blocks, and of course graduating and hitting those milestones. So students are my proudest moment overall. Absolutely, far beyond discoveries or naming organisms or anything like that.
Brandy and Lydia, thank you so much for joining us here on Absolute Genius. It was an absolute pleasure to have you.
Thank you for having us.
Yeah, thanks for having us.
That was Dr. Brandy Keel-Reese and Lydia Hayes-Guastea from the Dauphin Island Sea Lab at the University of South Alabama. Thank you so much for joining us for today's episode of Absolute Genius. Stay curious, and we'll see you next time. I'm going to do the whole Aspire ad in that voice. Being a scientist has its perks. Okay. Are we feeling aspirational? Are we ready?
Let's do it.